Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy in my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Kornelia Polyak. She's a professor of medicine at Harvard Medical School. She works in medical oncology. She's part of the Dana-Farber Cancer Institute as well. So we're going to talk about her research. Welcome, Kornelia.
Thanks for having me here today, and thanks for everyone for listening.
So tell me a bit about your background and how you started working in the cancer field.
I was born in Hungary. I grew up there, and I went to medical school because, we go there straight from high school. And I became very interested in cancer research and particular molecular biology, molecular genetics at high school. So pretty early on. So when I went to medical school, I started working in a biological research center, which is a kind of, Hungary is a small country, but it was kind of the institute that runs molecular research at that time. So this was in the 80s. And then I really wanted to pursue that. So I did come to the US and I had applied for graduate school at Cornell Sloan Kettering. And I worked with Joan Masagi there at Sloan Kettering. That was my PhD. And we identified P27 CDK inhibitor. And I just really, loved research. And I continued my training with Bert Vogelstein as a postdoc at Johns Hopkins, who's an amazing mentor, really I think the leader of founding father of modern cancer research. And then I decided, to focus on breast cancer and started my own lab at Dana-Farber in 1998. And we've been working on breast cancer ever since then. So that's how it was. So I was one of those unusual kids that, very interested in something from a young age. And I pursued it and I'm still fascinated by it. So.
Well, very good. What's the focus of your research, though, within the cancer world? Are you looking at it from an immunological approach, or what's your focus?
No, so we study breast cancer as a disease. So that includes all aspects of breast cancer. So we do work on trying to understand why women get breast cancer, what are the risk factors, and then why some people progress, others don't. With early stage breast cancer, what makes the progression, and we have studied the immune environment and the heterogeneity in those aspects. And then we also working on trying to identify drivers of the metastatic progression and also new therapeutic targets that we could better treat, particularly the highly heterogeneous tumors that tend to have worse outcome and they more likely to be resistant to current therapies. So that's how we call it. We don't work in any particular gene or aspects. We kind of study as a disease that allows us to look at everything that's important.
Well, is it one disease? I mean, I've heard from many cancer researchers that cancer is many different diseases. And within breast cancer, I know there's like triple negative. There's a bunch of different kinds. There's ductal. There's
Yeah.
So what does the landscape look like?
Yeah, so that's actually, we knew that from many years ago, but in the molecular era in the last like 2 decades became very apparent that even when we talk about breast cancer, it's not really one disease. Like we see these very major differences, like the three subtypes that, treated very differently: the hormone receptor positive, the HER2 positive, and the triple negative that's lacking all the hormone receptors and HER2. And those cancers, they really behave a different disease in a way. I mean, they have different risk factors for the incidence and progression, and also mutational profile and molecular profiles are very different. And they also, the treatment responses are very different. So in a way, yes, I mean, they all originate in the breast, but they are treated very differently. And we tend to focus more on the triple negative disease that is not currently having any approved targeted therapy beyond chemotherapy and immunotherapy for a subset of patients. But we're also very interested in, HER2 positive or hormone receptor positive disease that is resistant to treatment, the current therapies, which those breast cancer subtypes have more targeted therapies, like targeting the hormone receptors or the HER2 oncogenes. But unfortunately, a significant number of patients are still, recurrent, and they don't respond to treatment. And that's largely due to the heterogeneity of the tumors. And that's one of our main focuses, trying to understand why tumors are so heterogeneous, and then what drives the heterogeneity, and how can we improve treatment of highly heterogeneous tumors. So, I mean, that's how it's in a way, it's a multiple diseases under one umbrella.
When you talk about heterogeneity, how does that manifest? What kinds of heterogeneity are observed in tumors and in breast cancer in general?
Yeah, so we see heterogeneity that different individuals, different women have different types of breast cancer. That's kind of an inter-tumor heterogeneity. Then we see that even within one tumor at one time, not all cancer cells are the same. So they seem to have different properties, sometimes different mutations, like meaning genetic alterations. And then also when they progress, they continue to change. So I like to use the analogy of you have a bucket of balls of each, many different colors. And then that's kind of a tumor. You imagine those little balls are the cancer cells. And then the colors can change. There are those balls that kids have that you flashing and they change colors. So that's why it's so difficult to treat. Like you treating the green balls and then the red ones start to grow because they are lacking the target that you are focusing with the therapy. And then when the tumor progresses from the, local disease in the breast to the metastatic lesion, like imagine you have a bucket of balls and you randomly throw some of them into a different place. That's what kind of metastasis is. And then you start growing different tumors there. So, that's why it is heterogeneous, and particularly the advanced stage disease that is so extensive and have metastatized already. It's so difficult to treat because, you're treating, even if you're treating with 3-4 drugs, then, there is a subset of cells that is not responding, and then they recur and continue to grow. So that's why I think, very important to have early diagnosis, but also this is what we're trying to understand about this heterogeneity, like why some tumors are so heterogeneous, and how can we predict from at the time of diagnosis which patient is likely to be not responding to treatment and progress, and how can we treat them better? And obviously, based on, what we know about evolution and other cancer types or what we see in infectious disease, is that usually you want to use a combination of agents that have different kind of attacking the tumor from different ways. And it's just not so easy to work out those combinations. But, that's one of the areas that we and many others focus on, because that's what's going to make a difference.
Okay, so what is your overall approach to studying cancer then? It sounds, overall, and not not focused on one type of breast cancer, but where are you seeing that the headway can be made? What specific hypotheses are you testing in labs, or what are you working on specifically?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Yeah, so what we do is we treat cancer, look at cancer as a whole, look at as a not just the individual cancer cells, but, how they behave, because what we have been seeing is that, if you have cancer cells with different properties in the same tumor, the mixture behaves differently than each one individually. So we're trying to understand why that is and how can we target that. So we study clinical breast cancer. We study patient samples. We look at a very detailed molecular profiling, cellular composition of the tumors, and then we also study tumors as an intact tissue samples, because the location of the cells, relative location of the cells within a tumor is also important and determines many times response or risk of progression. And then even beyond that, primary tumors, as soon as they start growing, they start modifying the whole body in a way, so that even cancers that not yet progress to metastatic disease, they can have some systemic effects, particularly some that modulates the immune system, and then that makes some cancers more likely to progress. So we study that also, what kind of systemic effects the cancer has. But again, the areas that we're working on is study the heterogeneity, study what drives the heterogeneity, and how can we interfere that. So a lot of our work is studying the samples from the patients, and then we also develop experimental models that mimic that heterogeneity, and then we can do more functional studies to see how these mixtures of cancer cells behave differently than each one individually, and then what kind of treatment approaches can we apply to them that maybe make them more responsive. And then we're trying to translate those back to the clinic, particularly new agents, new therapeutic agents, and new combinations that we see seems to be more effective in the preclinical studies, and try to have clinical trials on those to see if they really work better in patients.
Okay, so again, what is the nature of heterogeneity? If we look at the mutation model and we see that the cells in the tumor have different mutations, would their surface membrane antigen presentation change? Would their localized microbiome change if they have one around them?
Yeah.
Would their cell-to-cell signaling change? What is heterogeneity, and how do you characterize it?
So, if you want to be very detailed, you could say that no two cancer cell is exactly the same even in the one tumor, because yes, you can have genetic mutational differences. They are in the different microenvironments, because some are more interacting with other cancer cells, others more interact with the stroma. The antigen presentation, that's exactly, whether they recognized by the immune cells or not, that is also variable within tumors. And then also some of them are, in breast cancer, for example, we know that some of them are more differentiated looking, like they look more luminal epithelium. Others look much more like a fibroblast, they're more mesenchymal. And then the ones that look more mesenchymal also tend to be more invasive, migratory, and more angiogenic, and also more immune suppressive. So they try to produce, they produce a lot of secreted proteins that kind of suppresses the immune cell function, or would not even let the immune cells to infiltrate in the tumors. For example, TGF-beta is one of those that, particularly has an effect both on kind of make a tumor less accessible to the immune cells, and also even if they enter, they makes them less functional. So I think all of those are modulating the heterogeneity. One particular area that we are very interested in is the epigenetic regulators. So like those genes that determine what cell type it is, those tend to be very commonly mutated in cancer, either amplified or actually have mutations. And the consequence of that the heterogeneity of the cells is increased. And the reason why we're interested in epigenetics, because epigenetic regulators and epigenetic profiles we can actually modulate. There are various agents in development that we could do that. So that way we could make the cancers look more homogeneous.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
And hopefully more homogeneous in a way that they would be responsive to a treatment. So if you imagine cancer cells can have 100 different phenotypes. And then if we treat with a epigenetic drug, then they go down maybe a few main pathways in phenotypes, and then they could become more responsive to treatment. So that's one of the areas that we're very interested in, and also published several papers to show that that's actually the case, that you have these mutant epigenetic regulators, and when they are mutant, then their tumors are more heterogeneous. And if we inhibit their activity, then we can make the tumors more homogeneous and make them respond to treatment better.
Well, if you're hitting the tumors with chemotherapy, as appears to be standard of care for most cancers, then that seems to drive heterogeneity. So how is anyone supposed to get a handle on this and fight it if chemo may or may not be used in a cocktail? And again, it appears to drive heterogeneity unbelievably.
That's actually not chemo is actually less biased than targeted therapy. Targeted therapy will always only hit the target and not the cells that lacking the target. So, for example, HER2 targeted therapy, which can be very effective, but, there are significant fraction of patients, we just published a paper on that lacking HER2. Within a tumor, you have 99.9% of the cells have HER2, and then you have this very small fraction, or some cases not even that small fraction, that is lacking the target, lacking HER2. So then it doesn't work. And chemotherapy, in a way, can be beneficial in cases like that, because chemotherapy doesn't require the presence of a particular gene. It's just targeting cells that are proliferating. So I think it depends on what kind of chemotherapy you approach, and also when are you applying. Because, for example, triple-negative breast cancer, almost half of the patients are cured by chemotherapy alone. The ones who respond, and, they cured really in a way that they will not have recurrence ever. Very, very rarely. Whereas, the hormone receptor positive disease, they're responding well to hormonal therapy, but then they can have very late recurrences. They continuously have recurrences. So I think it's not necessarily that chemo is bad. It's just depends on the tumor and depending on when you're applying. And certainly you don't want to treat people with chemotherapy that not likely to respond because you have an obvious side effects. But I think that's what we're trying to every one of us, including us, trying to achieve is trying to match the patient at the given time to the best therapeutic approach. Because, even the same patient at different stage of the disease or at diagnosis or recurrence or so, they may require different therapies. So, for example, in breast cancer, particularly in a triple negative disease, patients, when they diagnosed, very commonly undergo so-called neoadjuvant therapy, which is basically treatment even before surgical removal of the tumor. And it's usually lasts a few months. And during that time, you will see who is responding, who is not responding. In triple negative disease, almost patients who respond completely, 90% of them will not have recurrence. So, they essentially cured. And then there are patients, unfortunately, who don't respond. And then at that time, you could decide that how to treat them differently after surgery to not use the same kind of approach. So I think that's what we're trying to match that's what precision medicine is, in a way that we want to match the patient with the right therapy at the right time and then make changes as if that particular therapy doesn't work.
Well, what is the right therapy then? So it seems like you may be saying that some targeted therapies, if they get 99% of a tumor mass, the 1% is plenty enough for it to recur. You seem like you're saying some general chemotherapies, they're non-targeted, they work better. So how do you approach this then? Do you go for a targeted therapy combined with a general, or vice versa, or what order?
It depends on the tumor and depending on the treatment. So yes, so for example, in some cases, the targeted therapy is combined with chemotherapy or immunotherapy. Immunotherapy is kind of, also kind of not specific to a particular population. So combination would work the best. We just need to know which combination, which patient, and which tumor to apply. I know, there's many people say, we just need targeted therapy, but targeted therapy always selects for resistance. Every single tumor will recur that you're using only targeted therapy, especially if you're using only one type of a targeted therapy. So either you need to have a combination that targets all the populations and you have to have different targets, or you need to combine the targeted therapy with some non-targeted therapy that would somehow inhibit even the cancer cells that don't have the target. Or, the area that we are not as interesting is trying to understand why we have these heterogeneous populations. Like, for example, in HER2-positive tumors, why do we have HER2-negative cells? HER2 is supposed to be an important driver of those tumors. Why are they lacking? And then what are they doing there? As the tumor grows, why don't they just disappear? Because they seem to be growing slower. So then if we better understand that, then maybe we design different kind of combination for those patients. And whether that's going to be combination of targeted therapy with chemo or immunotherapy or epigenetic therapy, that depends on the particular case and at the given time. I think epigenetic agents will be very good combinations because they seem to modulate the tumor's heterogeneity. But again, those are still kind of just emerging agents, not many of them or very few of them are actually used in a clinic.
But what would epigenetic therapy look like? What would it do? How would it work?
Well, epigenetic therapy could be, some of it is used already, some of the differentiation therapies that used in some pediatric tumors and work amazingly, make cells differentiate. That's one of those. Others we and others working on that we know modulate the heterogeneity of the cell populations and trying to combine them with other agents. And we're talking about not just combining 2 drugs, but sometimes 3 or 4 drugs. And as long as they have different kind of ways of killing the cancer cells or targeting the tumor and not overlapping toxicity, I think that's the way it will work. That more combinations we apply, in general, we have higher efficacy. It's just we have to be able to do that safely. And again, not everybody needs it. People who can be cured, like early stage tumors or not that heterogeneous, smaller tumors that we can cure with one agent, we want to use one agent because why create more potential toxicities? So, but then others who are looking like they have a bad tumor, then they need more extensive combinations. And we need to assign that early on. And if you don't need it, then we don't need to need it. So it goes both ways. We don't want to overtreat people because that's always creates more issues long term. But at the same time, the people who have the bad type of tumors, we need to treat more aggressively and from early on because recurrences are much more difficult to deal with because, treatment itself is changing the tumor. You're selecting, obviously, when you're treating somebody, you're selecting for cancer cells that can continue to grow despite the therapy. So, of course, that changes the tumors very significantly. And we want to avoid that. Ideally, we want to kind of treat the tumor and kill as many cancer cells as quickly as we can.
Heterogeneity seems to be a necessary condition for tumor survival and for tumors to thrive. Even, it seems like, again, I may be wrong, but even when, a tumor, again, is attacked and there may be 1 or 2, clonal lineages that survive and then they proliferate again, but heterogeneity returns just in a different form.
Yes.
So I just wonder if heterogeneity is a necessary condition, again, for a tumor to survive. That's
heterogeneity, just coming back to the definition of evolution, what drives evolution? Heterogeneity and selection and reproduction. So tumors are the same. If you would not be heterogeneous and would be a very homogeneous population that will be killed by one drug, that the tumor would be effectively treated. But that's the challenge we have. And, so you treat the tumor and then you have a subpopulation that grows, and then that regenerates a different type of heterogeneity. And that's why it's so difficult to treat the, more advanced stage tumors and the recurrent tumors, because, that's evolution is very, 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 it's hard to stop. That's why, if we inhibit the proliferation, for example, the chemotherapy is in a way, if you slow down the proliferation of the cells, even though you may not cure them completely, but just slowing it down in combination with some other agent can be actually beneficial, because again, like you have selection, proliferation, heterogeneity are the three drivers of the progression. So modulating any one of them, or actually ideally the combination of them, is the best approach, because that's the most effective way of controlling the cancer.
If you look at primary tumors versus metastases, within breast cancer, what does the heterogeneity look like? How is it different in metastatic sites, or, pre or post chemo, or pre or post targeted therapy? Like, have people been able to characterize the type or flavor or color of heterogeneity? How is it different in the comparison?
Yeah. So, for metastatic disease, one of the obvious increase in heterogeneity comes from the different location of the tumor, because, a tumor that is growing in a brain or in the lung or in the liver, it's in a totally different environment. So there is a very strong selection for cancer cells that can grow in that environment. So you basically can have, in a diffusely metastatic patient that has metastatic lesions in multiple organs, one of the heterogeneity comes from that, you just have different locations and every one of them selects for different types of cells, and every one of them have different microenvironment, meaning like the surrounding cells are different. The immune cell ability to invade is different, like in the brain, they may not go in that well. So that comes from there. And at the same time, in breast cancer, almost all cases are recurrences after systemic therapy. So then you have the treatment itself can increase heterogeneity, sometimes in a way that you basically increasing mutation rates. Unfortunately, that can happen sometimes with some treatment. Other times you're selecting for a subpopulation of cells that is more resistant to treatment. So for example, in breast cancer, the hormone receptor positive disease that in general responds pretty well to treatment. But then when they recur, then they have mutation in the hormone receptor like ESR1. That's very common. Or many epigenetic regulators, interestingly, are more commonly mutated in the recurrent metastatic disease. For example, the ARID mutation, or there's this epigenetic complex that's very important for determining the epigenetic landscape of the cells, like what genes are active or inactive. Those tend to be more frequently mutated in the metastatic treatment-resistant disease, which will lead to increased epigenetic plasticity. So you basically selecting for cells that have many different features and increasing heterogeneity. And, that basically makes the treatment of metastatic disease even more difficult.
Well, what's your thoughts going forward on, the efficacy of treatments that you're working on and other scientists are working on? I mean, does it seem like it'll be close to breakthroughs specifically in regards to breast cancer or other cancers, or is it just, it's a big unknown? There's going to be decades more of work and who knows what will come of it?
I don't think it's decades. I mean, like the last, 5 years, we have amazing progress with immunotherapy in many cancer types that used to be, untreatable, like even lung cancer, the subset of patients that respond to immunotherapy, they're doing well. In melanoma, metastatic melanoma can be cured, a subset of cases, by immunotherapy or the combination of therapies. So breast cancer, unfortunately, is one of those cancer types that is not responding well to the currently available immunotherapies. But then the other thing, what we see is happening, that we need to apply those treatments at earlier stages, because as I mentioned, that earlier stages, the cancer is not less heterogeneous, and also you have fewer cancer cells. So, like even if you consider like a 1 centimeter cancer as a small cancer, that's billions of cancer cells, 10 to the 9, 10 to the 12 cancer cells. So if you imagine that having a subpopulation that's resistant to treatment increases the higher number you have, then of course, if you treat earlier, it's better. So, and also immunotherapy seems to be more effective in breast cancer when applied early, like in an adjuvant or even in a neoadjuvant setting. Of course, it's because cancer cells have not figured out a way to escape that immune system. And then the problem there comes is that some of those therapies have significant side effects, like immunotherapy can trigger an autoimmune disease that can kill you, or you trigger an autoimmune diabetes. So you're basically going to be diabetic for life. So there, one of the challenges is to figure out who's more likely to have a side effect, serious side effect, and at the same time, who is more likely to respond. So I think we have, that this better understanding of heterogeneity, but coming down, we need drugs. We need drugs from different types of genes and pathways. And then also we need more pipelines to evaluate combinations of agents and earlier stages, because the traditional clinical trial design is that when you're testing a new agent or new combination, you're testing them in patients who basically resisted every kind of therapy that you already tried. They really, really kind of refractory to treatment. And then you're asking a new agent that you're trying to test. Many times you have to apply as a single agent to cure a cancer that already resisted like 10 different ways of trying to kill it. So I think that also has to change. We have to be more open to move investigational drugs into earlier stages, because they may be more efficacious early on, but not at the end, when you basically trying to treat a refractory disease. And immunotherapy is one of the good examples for this, for breast cancer. The metastatic patients who already failed many therapies, only like 10-20% of them respond. But if you move it to early stages and combine it with some chemo, then you have 50% respond. So I think we need better drugs. We need better ways to combinations, better ways to assign patients to particular therapy. And also we need to change our clinical trial design in a way that take some earlier stage testing into the clinic more often.
Well, very good. Well, Kornelia, thank you for coming on the podcast. What's the best way for people to find out more about your work? Where can they go?
They can Google me and, the website. We have a lab website, or have email publications we have there, and they can check those.
Okay. Well, thank you for coming on the podcast, Kornelia. I appreciate it.
Okay. Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.